So the final class of non-coding RNAs we want to think about are the long non-coding RNAs. So as it suggests from the name, these are much longer than those short RNAs we were talking about before, the pi RNAs or the microRNAs. And in this case, they're now more than 200 nucleotides in length. In addition, according to their definition, they are usually spliced, so they undergo normal splicing like a regular messenger RNA would. They are capped, so they have a 5' cap, like a messenger RNA would, and indeed a poly-A tail, so they're polyadenylated. They also have the same sort of promoter as a regular gene would have, and so they're subject to expression in a developmentally controlled manner. But what's interesting about them is that they tend to be, or they should be constrained to the nucleus, at least predominantly. And this is looked at by using a method known as RNA fluorescence in situ hybridization. So you look at where the RNA is within a cell using the homology of a probe that can bind to that RNA in particular. And you want to see for a long non-coding RNA that the vast majority of that RNA is staying with the DNA in the nucleus. So the numbers of RNAs, the long non-coding RNAs that are found in the genome, the estimates vary dramatically. And so perhaps there's somewhere between 10,000 and 200,000 that are found in the mammalian genome. The vast majority of these are found at very, very, very low copy number. So there may be only a few copies of these long non-coding RNAs in each nucleus. So the question now becomes, what are these long non-coding RNAs doing? And are the ones that are found at such low copy really doing anything, or are they perhaps just the result of transcriptional read-through or transcriptional noise in the genome? What's interesting about the ones that have been characterized is that these long non-coding RNAs appear to regulate epigenetic processes. And this is what's interesting for them in relation to epigenetic control in this course. So these long non-coding RNAs can regulate many different processes. And as I said, there are a very large number of them. But there's a smaller set of them that have been characterized in any detail. We're going to talk about EXIST as well as hot air, and these two long non-coding RNAs have roles in exon activation for EXIST and Hox gene silencing for hot air. You'll recall exon activation, the dosage compensation mechanism in female mammals. It actually involves a very large number of long non-coding RNAs, but the prime example is EXIST, and this was indeed the first long non-coding RNA to be discovered back in 1991. There are many more that have been discovered since that have a role in exon activation, including T6. Genomic imprinting, which I introduced in the last lecture just with a couple of sentences, this monoallelic expression of genes that differs or is based on the parent of origin of the gene, also can involve long non-coding RNAs that have been very well studied. And Hox gene silencing involves hot air, which we will talk about, this long non-coding RNA called hot air. So Hox gene silencing, we have a large number of Hox genes in our genome, and these are involved in setting up the segmentation through the body axis. So for example, if you think of your vertebrae, so each Hox gene needs to be on in a particular region and then is switched off in other regions. And this switching off involves this long non-coding RNA called hot air. Finally, there are many other processes that are regulated with long non-coding RNAs, including DNA damage response. So what do these long non-coding RNAs actually do? Well, you'll remember one of these long-standing questions in the field of epigenetics that I brought up in a previous lecture was, how are the epigenetic complexes, so the complexes that are laying down DNA methylation or histone modifications, or that are moving around the nucleosomes, the chromatin remodelers, how are these actually targeted to specific sites in the DNA? We know that most sites actually don't have much DNA binding specificity. Most complexes don't have this DNA binding specificity all of their own. So we also know there are transcription factors, these factors that would bind with RNA polymerase, and they can provide some sequence specificity. However, the sequence specificity that's provided by a transcription factor tends to not be particularly high. So they may recognize maybe a 6-base recognition sequence or even a 12-base recognition sequence. But if you calculate the number of times that these 6 or 12-base recognition sequences can be found in the mammalian genome, it doesn't allow for unique targeting of any complex, but rather there'll be thousands of these sites spread throughout the genome. So finally, we also said that the chromatin remodeling complexes, for example, will be targeted based on previous histone marks, or they will bind to other histone marks. And we know also there are some epigenetic complexes that lay down a histone modification that also bind a previously made histone modification. But again, this doesn't allow for that specificity. We don't get to come down to just a few loci or one allele by using just these mechanisms. So the features of long non-coding RNAs somehow allow them to direct these epigenetic complexes, at least in some instances that we know about at the moment. And they appear able to do this in cis, so in other words, on the chromosome from which they are transcribed, or in trans, so acting somewhere else. They do this amongst other functions. There are many functions that we'll come to, and we're just going to go through these ones that are concentrating on them directing the epigenetic machinery. So what are the features that allow them to be involved in this targeting, and especially to allow this allele specificity? Well, because they are transcribed, while they're being transcribed from a particular locus, from a particular chromosome, they are still tethered there. They're still tethered to the place from which they're being transcribed. And so this allows for that allele specificity. And it also shows how they might act in cis. Now, the reason they can provide specificity that's greater than that of a transcription factor is simply because of their length. Because of the length of the molecule, the length being over 200 nucleotides in length, and in fact, most long non-coding RNAs about which we know something at the moment are several kilobases or even tens of kilobases in length. So this means thousands of bases, of course. This really gives unparalleled site specificity for these long non-coding RNAs. But it's important to note that this specificity and to the allele, or specificity based on the sequence, is not necessarily always required for their function. So what are some of those functions? What are some of the mechanisms by which they work? The two that we'll focus on are the long non-coding RNAs acting as guides, and the example that we'd like to talk about here is EXIST. And we'll also talk about how hot air acts as a guide. The difference being that EXIST acts in cis, whereas hot air acts in trans, and I'll go through this in more detail. We know that these long non-coding RNAs can also act as scaffolds, and again, hot air is the example here. But in addition to these two mechanisms, there are many others that long non-coding RNAs, many other mechanisms by which long non-coding RNAs can function. They can act as decoys, they can act as signals, and what this means is that it's actually the act of transcription itself running through a locus which has the function rather than the product, rather than the long non-coding RNA that's produced. They can act as enhancers, and this has always been shown to work in cis, so you can have transcription at one point which alters transcription nearby. And they can even act as reservoirs. And in this context, H19, which is one of the best characterised long non-coding RNAs, acts as a reservoir of a microRNA. So it can sit around being a stable long non-coding RNA that can then be processed to release the microRNA that it contains. So while there are so many different ways that these long non-coding RNAs can actually work, there is a common feature, and that is that there's the formation of an RNA protein complex. And this RNA protein complex is able to influence gene expression. So I'll just briefly describe by what I mean as when things act as a guide versus a scaffold. And then in the next lecture, we'll go through an example of each of these. So as a guide, what I mean is that this epigenetic complex, which I've drawn here, will bind to the non-coding RNA. The non-coding RNA is drawn in red. And so we have this long non-coding RNA coming along, and it is tethering an epigenetic complex to a particular region. The reason that it's site-specific is that there's sequence identity, which is allowing this RNA to bind to the DNA in a sequence-specific manner. In other words, the long non-coding RNA is guiding the epigenetic machinery. This epigenetic machinery can then participate in whatever it normally does, in this case, histone methylation. So in this way, one complex is being guided to a specific site in the DNA. However, when these long non-coding RNAs act as a scaffold now, rather than just recruiting one complex, they tend to recruit more than one, in this case two. So different regions of the long non-coding RNA, so this region here or the region down here, combine to two different epigenetic complexes. And so in this way, they act as a scaffold They unite two complexes so that they are tethered to one another and act as a molecular scaffold to bring epigenetic complexes somewhere else. This scaffolding may not necessarily require that they that their long non-coding RNAs provide sequence specificity. But you have then got tethered together these two different complexes which are then able to act on chromatin and at the same location, tethered by these scaffolding long non-coding RNAs. So in the next lecture, we'll talk about examples of each of these cases and go through their particular functions.